This is Peter Brown from Bio 108, and this is the lecture on DNA to protein as well as viruses. So DNA is the information of life, and the information in DNA tells the cell how to build proteins. So the recipes in DNA that tell the cell how to build proteins are genes. And DNA has two kinds of information. One kind of information is just how to replicate itself. So every time a cell divides, it needs to replicate the DNA prior to cell division. So DNA is a double-stranded molecule, and the two strands separate, and each strand gets replicated. So when a cell divides, every new cell has identical DNA to the original cell. The other thing that DNA does is DNA has the information on how to build proteins. Those are genes. And so there's sort of an intermediate step where DNA builds RNA and the RNA builds proteins. And sometimes people say this is the central dogma of biology, or the central idea that all living things have information, and that information is DNA, which tells the cell how to build proteins. So when a gene is turned on, DNA is being used to make RNA, and then that RNA is used to build the protein. So here's an enzyme in brown, and the enzyme is called RNA polymerase, and it's copying a gene on the DNA and building RNA. And then the last step is the RNA to protein, and this blue thing is a ribosome, and the RNA gets latched onto the ribosome, and it builds a sequence of amino acids, which are the building blocks of proteins, and those amino acids will wind up into a functional protein. Okay, this is a great video that gives an analogy of a chef making proteins. So if you watch this video, the first part is the DNA to RNA to protein analogy, and then the second part talks a little bit about viruses. So by watching this video, you'll kind of get an idea where we are and where we're going with today's lecture. All organisms share a common ancestry, and one of the most compelling pieces of evidence that this is the case is that DNA is the same in all organisms. So that's sort of just a more abstract way of thinking about it, but a more practical way of thinking about it. Because DNA is the same in all organisms, we can do genetic engineering. So people who are diabetics these days, most of them don't get their insulin from pigs' pancreases, which is where they got them from 50 years ago. Instead, most of them have recombinant DNA that's grown in E. coli. They took a gene from a person and put it into an E. coli bacteria, and then that E. coli bacteria spits out human insulin. It's not something that's like human insulin. It is human insulin. And E. coli reads its DNA recipe the exact same way we do. And when you put that gene from human into E. coli, the bacteria spits out human insulin. This is a genetic engineering lab that we do in our Bio 100 class, or we have done it in the past. I haven't taught the class in a few years, but it's basically using bacteria, and they take a gene from jellyfish that glows in the dark, and they put it into bacteria, and to see if it got into the bacteria, you put a ultraviolet light, a black light on, and if it glows in the dark, we know we got the gene in. Okay, let's switch from DNA to viruses. And so viruses are really under the microscope these days, both figuratively and literally, because of COVID-19. The first scientist to find a virus was a Dutch biologist whose name was Martinus Beijerinck. If you know how to pronounce that, you can let me know. Anyways, he found a substance infecting tobacco leaves in the late 1800s, and he found that filters that filtered cells didn't filter this particle. So we didn't know what it was, but today we know it's about 10 times smaller than a bacteria, and it's a virus, and here's the tobacco mosaic virus seen with an electron microscope. So what are viruses? So this is a really nice quotation from Concepts of Biology. No one knows exactly when viruses emerged or where they came from. There's no historical footprints such as fossils, but modern viruses are a mosaic of bits and pieces of nucleic acids picked up from various sources along their evolutionary path. Viruses are acellular. Acellular means they're not made of cells. They're parasitic entities that are not classified within any domain because they're not alive. No cells means no plasma membrane, no organelles, no metabolism, no division. Instead, they infect a host cell and use the host cell's replication process to make more virus particles. So viruses replicate, but entirely from stealing the host cell's metabolism. Okay, so there's a bacteria and there's a flu virus 10 times smaller. Okay, so here's just comparing a bacteria cell with a virus. So bacteria are cells, and viruses have a protein shell that contains DNA, or sometimes it contains RNA. Okay, bacteria are 10 times larger, viruses are 10 times smaller. Bacteria reproduce by cell division. Viruses infect host cells and use host cells to make more viruses. Bacteria can move by flagella, or most can. Viruses only move passively, for example, by sneezing and being suspended in the air. Antibiotics will kill bacteria cells. We know their mechanism, and different antibiotics have different mechanisms. So, for example, penicillin kills bacteria by preventing them from building cell walls. And viruses, the best way to kill viruses is by vaccination, which causes the immune system to recognize the protein from the viruses. And so if you get that virus, your immune system recognizes the virus and responds. Examples of some bacteria that cause infections are strep throat, having a urinary tract infection, tuberculosis. Okay, and examples of viruses are things like the flu, getting a cold, or HIV, the virus that causes AIDS. So you can see they're not all the same. Some spread relatively easily, like the flu and a cold. Others, to get HIV, you'd have to share a needle or have unprotected sex. All right, here's just a video showing, a diagram showing how a flu virus invades a cell and uses a cell to make more viruses. So here's a virus. You can see the cell is much larger. There's a receptor on the virus that the cell responds to, and then the cell thinks the virus is something friendly, so it takes it up into the cell. Once the virus gets into the cell, it uses the cell's machinery to make more viruses. Those viruses are assembled, and then that virus will leave the cell, and it can infect new hosts, new cells. Okay, lastly, at the beginning of the semester, we saw a video on vaccination and what it was all about. And I'm going to have you watch it one more time and answer the quiz questions. This is a video on smallpox, which is unusual that the virus has no animal hosts that get smallpox. There's a similar disease called cowpox, which is found in cows, but there's no smallpox in animals. other than humans. Because of that, we've actually been able to wipe smallpox out. That's not true for things like flus because there are avian or bird reservoirs. Even if we wipe the flu out of humans, birds would still have it and then give it to humans.